So pharmacokinetics properties is only considered for drug molecule, okay, not for the proteins. So in our last class, we performed docking between ligand and protein, and we suggest a compound as a top-ranked molecule, and this could be a candidate drug molecule against that protein. So if we consider this one as a drug molecule, then this compound should have some properties like a drug. Okay, so that's why we consider this session for the calculation of pharmacokinetics and other physicochemical properties of any molecule. Uh, Before considering any compound as a drug, or before suggesting any compound as a drug molecule, um, we have to perform this analysis, and we have to consider their pharmacokinetics and physicochemical properties as well as toxicological properties. Okay, are they toxic or not? Uh, First we will talk about drug likeness properties, and these drug likeness properties are the qualitative concept, okay, that is used in drug design to calculate its druggable properties. That uh, these properties calculate some physicochemical properties from its molecular structure, and it's only considered for the bioavailability. So if these compounds are bioavailable or not, so it is estimated from the molecular structure as I mentioned, and before the substance is even synthesized or tested. So if we are planning to synthesize any compound, it is better to design it in computationally and then calculate its properties, that is it a drug-like molecule or not, and we may consider some other properties, that is it will be suitable or not. Okay, so there are lots of molecular properties that we can calculate, but we will consider some properties, for example, structural properties, their physical chemical properties, and as well as biological properties. So, in terms of structural properties, we will calculate the special type of bonding, for example, hydrogen bonding, polar surface area of any compound, and its lipophilicity, their shape and geometry, molecular weight, reactivity, pK value. And in case of physicochemical properties, we will calculate its solubility, uh, especially the permeability, it could be a cell permeability, um, and chemical stability. And in terms of biological properties, we can calculate the metabolism, for example, in the phase 1 and phase 2 metabolism reaction, and then protein and tissue binding. It is the part of distribution and transport. It also the part of distribution, that after absorbing your compound by your body, then how it will distribute to the cell or tissue. Um, and then pharmacokinetics properties and their toxicity, for example, clearance and half-life. Clearance and half-life are the part of excretion properties that describe the properties that how your drug molecule could be ejected from your body after metabolism process, and also the bioavailability. Okay, so for the drug likeness properties, to calculate the drug likeness properties, or before considering any compound as a druggable or drug-like molecule, we have to consider some properties, and for this, we have some methods like Lipinski's rule of five. So Lipinski's rule of five is one of the most leading method to calculate the bioavailability of our. Okay, so the Lipinski rules provide us some assumptions that by the violation or if the compound violated the Lipinski's rule, then we cannot say that this could be a drug-like molecule. If the molecules obey the rules of Lipinski's, then it could be a drug-like molecule. So let's discuss on it. Okay, Lipinski's rule of five proposed by Dr. Christopher Lipinski is a set of guidelines used in drug discovery and development to assess the drug likeness of chemical compounds. These rules are based on empirical observation of the physicochemical properties of orally administered drug. The rule helps in predicting the likelihood of a compound success as an orally active drug. So, Lipinski rule is only considerable for the orally administered drug. So you can check the properties that if your compounds are orally active or not. Okay, if you are designing any compound such as ointments or any compound that should be taken by your vein, so for that case, you not need to obey the all properties of Lipinski's. So Lipinski rule of five consists of the following criteria, molecular weight. Lipinski consider the molecular weight and the lipophilicity value, or sometimes it could be considered as a log p value, or it also referred as the water octanol partition coefficient sometimes, according to chemistry, and then hydrogen bond donor and hydrogen bond acceptor. So what are the standard range of these properties, right? In case of molecular weight, the molecular weight not greater than 500 Dalton. So it should be less than 500 Dalton. And the log p value or lipophilicity should be less than or equal to 10 or equal to 5. And hydrogen, in case of hydrogen bond donor or HBD should be not more than 5, and hydrogen bond acceptor should not be more than 10. So these are the rules of Lipinski. Okay. So if any compound just violate one one rule, then you can consider this compound as acceptable rate for the orally active, or you can say that your compounds are orally active and your compounds obey the Lipinski's rule. But if your compound violate two or more than two rules, then you cannot say that your compounds are okay, your compounds are drug-like, or your compounds are orally active. So, is it necessary to maintain the Lipinski's rule? And the answer is no. It is neither the qualitative nor foolproof. Okay. For example, orally active drugs such as these drugs do not obey the rule of thumb or the five. So, rule of thumb and the Lipinski's rule are the same. In case of large molecular weight compound, invariably have too many functional groups. Okay. Why Lipinski consider the molecular weight for 500 Daltons? Because if your molecules has the larger weight, then there are the chance to have too many functional groups, and these functional groups can be form hydrogen bond. Okay. And GSK and GlaxoSmithKline rule is another rule for considering a drug likeness molecules. You can consider these rules also, but the acceptance of Lipinski's rule is higher than GSK. Okay, so according to GSK rule, molecular weight should be less than 400 Dalton, and hydrogen bond donor should be 7 or less than 7. Hydrogen bond acceptor should be 10 or less than 10, and the log p value should be less than 4.5. Okay, so here's another popular rules, Weaver rules, which is mostly acceptable rules after Lipinski, and these rules have four properties: hydrogen bond donor, acceptor, a newly added rotatable bond, and polar surface area. So the hydrogen bond donor and acceptor should not more than 12 total. Okay, total number of hydrogen bond donor and acceptor should not be more than 12 or equal to 12. And the rotatable bond, the number of rotatable bonds should not be more than 10. And in case of polar surface area, the polar surface area of any molecule should be less than 140 angstrom square. Okay, and the unit of polar surface area is angstrom square. So what is the assumption was that was carried by Weaver and his co-workers. So they find that molecular flexibility as measured by the number of freely rotatable bond. So molecular flexibility as measured by the number of freely rotatable bonds. So the number of freely rotatable bond and flexibility are same. So they find that molecular flexibility plays an important role in oral viability, and the more flexible the compound or the more flexible the molecule, the less likely it is to be orally active. So if your compounds have more rotatable bond, or if your compound has a higher rotatable bond than others, then there is a chance to be orally inactive. Okay, so less surprisingly, the analysis showed that less surprisingly. Okay, so this is the second findings. This is the main findings of the study, and this is the second finding of this study. And they find that the analysis showed that the popular surface area of the molecule could be used as a factor instead of the number of hydrogen bonds. Okay, so in our previous methods such as GSK or Lipinskis, they consider hydrogen bonding groups, for example, hydrogen bond donor and acceptor. But Weber says that uh, their analysis shows the polar surface area can be replaced. Uh, okay, so we can consider the polar surface area instead of hydrogen bonding groups, and the standard range for acceptable oral activity of any compound. So a polar surface area less than equal to 140 angstrom square. Uh, Sorry for the unit mistake, and less than equal to 10 rotatable bond. Or if we consider the hydrogen bond groups, then the total number of hydrogen bond donor and an acceptor should be less than equal to 12, and less than equal to 10 rotatable bonds should be considered. Some researchers set the limit of rotatable bond less than equal to 7. Okay, since the analysis shows a marked improvement in oral bioavailability for such molecules. So if you reduce the number of rotatable bond to 7 from the 10, then the analysis shows that there will be a good improvement there in oral bioavailability. Okay, so these rules are independent of molecular weight, so there is no limit for molecular weight, and open the way to studying larger structure that have been solved up to now. Okay, so this is a good chance. So what are the hydrogen bond donors? So I think most of the student in our classes are from the pharmacy background, and they have no enough strong chemistry knowledge. So that's why I'm considering these topics for the discussion, like hydrogen bond donor. What are the hydrogen bond donor, and what are the hydrogen bond acceptor, and what is rotatable bond or not. So this will clear your knowledge about the drug likeness properties. So hydrogen bond donors are the groups which possess a hydrogen bond, or which possess a hydrogen atom bonded to a highly electronegative atom. For example, oxygen, nitrogen, fluorine, or chlorine, or bromine. Okay, the electronegativity difference creates a partial charge on the hydrogen atom and making it slightly acidic. Okay, so this positive charge can attract the lone pair of electron of an hydrogen bond acceptor, forming a weak electrostatic interaction called a hydrogen bond. For example, uh, hydroxyl group. So hydrogen is bonded here with the oxygen amino group, okay, and thiol group. Hydrogen is bonded with the sulfur. For example, these are in cysteine or some other drugs, sulfur drugs, okay. Hydrogen bond acceptor. Hydrogen bond acceptor contain lone pair of electron on highly electronegative atom. For example, oxygen, nitrogen, fluorine, chlorine. So oxygen, nitrogen, fluorine have lone pair of electron, and this lone pair of electron are negatively charged and can be attracted to the partially positive charge on hydrogen bond donor, as the hydrogen bond donor possesses a hydrogen bond with a highly negative atom. So the partially positive hydrogen uh, can be attracted by this lone pair of electron of hydrogen bond acceptor and form a hydrogen bond. Some example of hydrogen bond acceptor groups: carbonyl groups, okay, ether groups, nitrogen lone pair available in pyridine, amine, and other nitrogen containing molecules. So the importance of hydrogen bonding, we can calculate the water properties, okay, the heat capacity, surface tension, and solvent properties. Protein structure and function. Hydrogen bond stabilize the protein folding and interaction with other molecules. In drug development, understanding the hydrogen bond donor and acceptor group help design drugs that interact effectively with biological targets. So remember these topics. The understanding hydrogen bond donor and acceptor groups helps design drug that interact effectively with biological target. We will discuss this particular point in later slide. Okay. Then what is the rotatable bond? Okay, rotatable bond are single bonds in molecule that allow for free rotation around the bond axis. This means that two connected atoms can freely rotate relatively to each other and significantly impacting the molecule's shape and flexibility. So here I denote the rotatable bond, and if we rotate this atom and this atom according to this bond axis, then these compounds form another conformation. Okay, that's why significantly impacting on its molecular shape and flexibility. So in our last class, when I showed a video in the slide, uh, I don't know, are you guys remembered or not? The molecules searching a position for better binding, right? By changing its conformation using this rotatable bonds. Okay. So, what are the importance of rotatable bonds? Okay. In case of structure and flexibility, rotatable bond can help in protein folding, drug target interaction. Okay, the rotatable bond can enable drugs to adjust their shape to fit into specific binding pockets on target molecule. Okay, in case of enzyme catalysis, rotatable bond in enzyme allow them to move substrate molecule into optimal position for chemical reaction. I don't know that you have remembered or not about the slide induced fit docking, where enzymes or proteins could be induced by changing some temperature. Okay, so they can change their conformation due to the rotatable bond. And in case of drug discovery, membrane permeability, rotatable bond can affect how easily a drug can pass through the cell membrane. Okay, molecules with fewer rotatable bond are generally considered more rigid and can traverse membrane more rapidly, and leading the better absorption. So, and another term, solubility and stability. The number of rotatable bond can influence the molecule's solubility and stability in various environments. Drugs with fewer rotatable bond tend to be more water soluble and stable, which is desirable for oral absorption. So the polar surface area. Polar surface area, or the topological polar surface area, PSA and TPS, are the same thing of a molecule, is defined as the surface sum overall polar atom. So this is nothing than the hydrogen bond donor and acceptor. So if we calculate the total polar atoms on the surface of a molecule, and then the result we consider as the polar surface area. So primarily oxygen, nitrogen, and also including their attached hydrogen atoms are considered for this calculation. And this is the surface area of a molecule occupied by polar atoms, for example, nitrogen and hydrogen, and functional groups like hydroxylamine and carbonyl groups. And Weaver rule suggests that a maximum polar surface area of 140 angstrom for good oral bioavailability, and high polar surface area can lead to increase the water solubility, pore membrane permeability. Okay, so if your compound have a high polar surface area, okay, it could be a good water soluble compound, but this will not uh, be a good membrane permeable compound. Okay, and hindering the absorption in the gut. And for a molecule to be penetrate the blood-brain barrier, if your molecule need to be penetrate the blood-brain barrier, then and if your compound have to interact with the central nervous system, then the polar surface area of your compound should be less than 90 angstrom square. Okay, this is the acceptance uh, range for the only for the blood-brain barrier cases. Okay. So let's start with pharmacokinetics properties. So what is pharmacokinetics properties? Pharmacokinetics properties is the quantitative study. Okay, pharmacokinetics is the quantitative study of drug movement through, in, through, and out of the body. That's how your compound will be absorbed, how this absorbed compound will move throughout the body, and after absorption and distribution and metabolism, how your compound uh, will be excreted from the body. Okay, the intensity of the effect is related to the concentration of the drug at the site of action, which depends on its pharmacokinetics properties. And pharmacokinetics properties of a particular drugs are important to determine the dose, the route of administration, onset of action, peak action time, duration of action, and the frequency of dosing. Okay, so the four main components to consider in pharmacokinetics are absorption, distribution, metabolism, and excretion. So let's discuss one by one. So absorption is the transfer of a drug from its site of administration to the bloodstream, and most of the drugs are absorbed by the way passive transport, and intravenous administration has no absorption, and the fraction of administered dose and rate of absorption are important. There are several factors that can affect the absorption. For example, molecular weight, lipophilicity or log P, ionization, solubility, permeability, route of administration, fast pass metabolism, and drug formulation. So, how molecular weight affect the absorption, right? So, smaller molecules are generally more easily absorbed. Lipinski's rule of 5 suggests that the molecular weight uh, should be less than 500 Dalton, and it is desirable for the oral bioavailable compound. And in case of lipophilicity or the log p value, so lipophilic compound means that it's tend to absorb by the lipid, right? So These compounds are lipid soluble or fat soluble. So lipophilic compounds are generally absorbed more efficiently, and however, an excessive degree of lipophilicity can lead to poor water soluble, uh, as our blood contains more than 70% of water. Okay, so that we have to consider a standard range for lipophilicity, so not higher or not too much lower lipophilic compound. Okay, ionization, the degree of ionization of a drug at a specific pH affects its absorption. So the ionization properties of any drug can control its ionization process. Okay, that's for example, if your drug need some weak acidic or acidic environment to ionize, then stomach will be the best for the ionization. And if your compound need a basic environment for its ionization and absorption, then the small intestine with pH environment are higher than 7. It could be up to 7.4, 7.5. Okay, the standard range for small intestine pH are maybe 7.4. And the solubility, water solubility is crucial for dissolution of a drug in the gastrointestinal fluids, but poorly soluble compounds or poorly soluble drugs may have limited absorption. Permeability, the ability of a drug to cross the biological membrane, for example, the cell permeability, is a key determinant of absorption. And Caco2 cell permeability assays are commonly used to predict uh, oral absorption. Route of administration. So the absorption profile varies with the route of administration, for example, oral or intravenous or intramuscular, etc. So GI absorption or gastrointestinal absorption is one of the properties that calculate for absorption. So gastrointestinal absorption or GI absorption refers to the process by which substance, including drugs and nutrients, are absorbed from the gastrointestinal tract into the bloodstream. And the gastrointestinal tract is a complex system comprising the stomach and the small and large intestine. So, here various mechanisms contribute to the absorption of substance. GI absorption is critical step in pharmacokinetics for orally administered drug and the uptake of nutrients from the ingested food. So, importance in drug discovery, bioavailability and therapeutic efficacy. Efficient GI absorption is crucial for achieving the therapeutic concentration of drugs in bloodstream and leading to desired therapeutic effects. And there are some factors that influence the GI absorption: pH environment, solubility, particle size, blood flow, and the surface area. Now, the distribution is the second phase in ADME process, okay, which describes uh, the fate of a drug within the body after it has been absorbed into the bloodstream. The distribution involves the movement of a drug from the bloodstream into various tissues and organisms, and it plays a critical role in determining the drug's concentration at its site of action. Okay? So, the blood flow, tissue permeability, plasma protein binding, and drugs characteristics, for example, molecular weight, charge, lipophilicity, influence, can influence the drug distribution process. Okay, blood-brain barrier is one of the key parameters for distribution, and the blood-brain barriers or BBB or triple B is a highly selective semi-permeable border of endothelial cell. Okay, that regulates the transfer of a solid and chemicals between the circulatory system and the central nervous system, thus protecting the brain from harmful and unwanted substance in the blood. So we can consider a blood-brain barrier as a international border between two countries. Okay, as they check some unwanted peoples who do not have any visa, okay, or who have any criminal activities. So like this, blood-brain barrier negotiate with chemicals before um, approve him to enter the blood-brain barrier. Okay, so the blood-brain barrier is an important barrier that drug have to be negotiated if they are to enter the brain, and the blood capillaries feeding the brain are linked with tight fitting cells which do not have contain pores, unlike capillaries elsewhere in the body, for example in others intestine. Moreover, the capillaries are coated with a fatty layer. Okay, so the capillaries are coated with a fatty layer and formed from the nearby cell and providing an extra fatty barrier through which drug have to cross. Therefore, drugs entering the brain have to dissolve throughout the cell membrane. As our cell membrane are fatty layer, so your compound have to be fat soluble. Okay, that's why polar drugs such as penicillin do not easily enter the brain. Okay, uh, as the blood barrier are coated by a fatty layer, and if our compounds need to be enter the brain, then we have to pass the blood-brain barrier. So if our design compound uh, are the polar, then we have to manage this polarity to cross the blood-brain barrier. So how can we do that? So let's start with some examples. Okay, where we need to cross the blood-brain barrier, and where not. So if you design any compound or any drugs that have to be interact with brain and the central nervous system, then only then your compound should pass the blood-brain barrier. Otherwise, not. If your disease have no activity with brains, and if your compound have no activity with the central nervous system, then if your compound can pass the blood-brain barrier, there will be a risk for the central nervous system. Okay, so the existence of blood-brain barrier make it possible to design which will act various part of the body. For example, you can consider the heart and have no activity in the brain, thus reducing the central nervous system side effects. And this is done by increasing the polarity. Okay, as we know that polar compounds cannot easily cross the blood-brain barrier. So if your compounds uh, cross the blood-brain barrier, you can just redesign it as a polar compound. You can edit its certain group, and you can put some polar group to increase its polarity. Okay, such that it does not cross the blood-brain barrier. On the other hand, drugs that are intended to act in the brain must be designed such that they are able to cross the blood-brain barrier. And in this case, if your compounds uh, are polar, then we know that polar compounds cannot cross the blood-brain barrier. Okay, so this means that they must have a minimum number of polar groups and have these groups temporarily masked. So having said that, some polar groups can cross the blood-brain barrier with aid of carrier proteins, and others can cross by the process of pinocytosis process. Okay, previous we do not just we just remove this slide of pinocytosis. And the ability to cross the blood-brain barrier has an important bearing on analgesic activities. Okay, so blood-brain to calculate the blood-brain barrier, boiling method is very important. So we can see that the graph is look like a boiled egg, right? This is the yellow zone of your egg, and this is the white zone. So this plot is considered for the log p value and the polar surface area. Okay, based on polar surface area and the log p value. Okay, if your compounds are within this region, within this yellow region, then this means that your compound can cross the blood-brain barrier. Okay, here is the legends. And if your compound are in the white region, then this means that your compound can pass the other intestinal. Okay, and it has a good absorption property, and it cannot pass the blood-brain barrier. So this molecule is considered for the neurological disorder, and we know that if we design any drugs for neurological disorder, then this drug should have to uh, cross the blood-brain barrier. And again, these two compound are considered for other cardiovascular disease, or you can that have no action for our brain. And we can see that these two drugs are not within the yellow range, and these compounds have no relationship with the blood-brain barrier. And the third part of ADME is the metabolism, and compounds begin to break down as soon as they enter the body, and the majority of small molecule drug metabolism is carried out in the liver by redox enzyme termed cytochrome P450 enzyme. Okay, so this redox enzyme are also known as the cytochrome P450 enzymes. As metabolism occurs, the initial or the parent compound that we consume is converted into new compounds called metabolites. Okay, it's converted into several fragments, and these fragments are known as the metabolites. When metabolites are pharmacologically inert, okay, after converting to metabolites, if your metabolites are pharmacologically inert, metabolism deactivates the administered dose of parent drug, and this usually reduces the effect on the body. Okay, metabolites may also be pharmacologically active or inactive, or could be toxic. Okay, so if your metabolites are pharmacologically active than the parent drugs, this could be termed as prodrug. There are some important notes for metabolism process. There is a process detoxification. The primary purpose of drug metabolism is to convert potentially harmful xenobiotics, for example, the foreign substance like drugs, into water soluble metabolites that can be readily excreted from the body through the urine. Okay, this process typically involves with two phase 1 reaction and phase 2 reaction. In phase 1 reaction, this phase 1 involves enzymatic modification like oxidation, reduction, and hydrolysis process. Okay, so resulting in more polar metabolites. So the phase 1 uh, generates polar metabolites, and in phase 2 this involves the conjugation with various endogenous molecules like glucuronic acid or glutathione, further increasing water solubility for excretion. Okay, so for the excretion properties, your compound should have a good water soluble properties. Okay, and should be polar. And another process is known as the active inactivation. In most cases, drug metabolism leads to inactivate or inactive metabolites, effectively lowering the active drug concentration in circulation and consequently in pharmacological effect. This determines the duration of action of a drug. And another one is activation and toxicity. In case of activation, your drug metabolism process can convert a prodrug into its active form, and this allows for target delivery and controlled activation of drug at the desired site of action. And toxicity in certain situation, uh, metabolic byproduct can be more toxic than the parent, okay, leading to some adverse side effects. Okay, understanding the metabolic pathway of a drug is crucial for identifying potential toxicity risk. So this is the total metabolism process we discussed in our previous slide. So phase one, enzymatic detoxification pathway, then active metabolites, and okay, others. So in case of activation, so as soon as possible your compounds converted into metabolites, and then the excretion methods activate. Okay, so excretion is a process in which metabolic waste is eliminated from an organism, and in vertebrates this is primarily carried out by the lung, kidney, and skin. Okay, for example, for human, okay, unless excretion is complete, accumulation of foreign substance can adversely affect normal metabolism, and there are three main sites where drug excretion occurs. The kidney, the kidney is the most important site, and it is where products are excreted through the urine. And the biliary excretion or facial excretion is the process that initiate in the liver and passes through the gut until the product are finally excreted along with the waste products or phases. Okay, the last main method is of excretion is through the lung, for example the anesthetic gases. So is the schematic representation of your excretion process by kidney. Okay, and the kidneys are the principal route, as I mentioned in our last slide, that this is the major parts by which most of our metabolites can excreted. So excretion of drugs by kidney involves three main mechanisms. Okay, glomerular filtration, and then active secretion, and then filtered 100-fold concentrated in tubulars or for favorable concentration gradients. Okay, okay. The last topics is the toxicity. Obviously, we have to calculate the toxicity of any compound before we are going to lab work. For example, if we are considering to synthesis any compound, we have to first calculate its toxicity. Okay, so sometimes the potential or oral toxicity of the compound is taken into account. Okay, and this all are considered as the ADMET. Okay, A for absorption, D for distribution, M for metabolism, E for excretion, and T for toxicity. Parameters used to categorize toxicity includes the median lethal dose and the therapeutic index. And computational chemists try to predict admitox qualities of compound through methods like QSPR or QSAR or in vitro assays. So there are some topics uh, with the type of toxicity, and we added here some special type of toxicity like acute toxicity, chronic toxicity, subchronic toxicity, idiosyncratic toxicity. Okay, and the LD50 and LC50. LD50 are the lethal dose. Okay, and LC means the lethal concentration. So lethal dose can be defined as the dose. Okay, for at which 50% of your experimental animal will be expected to die. And the LD50 is one way to measure the short-term poisoning potential. For example, acute toxicity. Okay, For inhalation toxicity, air concentration are used for exposure values. Thus, LC50 or lethal concentration 50 is used otherwise, and LD50 is units are milligram per kg, okay, or unit or pound, okay, and LC50 as it is concentration, so milligram per liter, okay. So milligram per liter is estimated air concentration of a substance as administered via the inhalation route. Okay, sometimes it can be calculated as the ppm. Okay, so I suggest some tools that can predict the admit properties. First, the swiss admin pkcsm, deep pk. Deep pk and pkcsm are the web tools, and deep pk is belong to the pkcsm. Admit lab 2.0 admit serve. And for calculating the toxicity, we can consider the protox. Okay, so as the end of your theory session, if you have any question you can, otherwise we will go for the practical session. Okay, let me close the slide and let us create a folder here for class 6. Okay, so to calculate the admit properties and physicochemical properties of any compound, we need a smiley format. Okay, so in our first session, I suggest you that to collect a smiley in Excel sheet, right? So let's collect some smiley from PubChem database. Okay, we may consider the smiley list from here. Okay, here I have some smiley. Okay, one, two, three, four, five. Okay, I have five smiley. Okay, one, two, three, four, and five smiley. So for this study, as example, I consider only five compounds and just copy this smiley. Okay, now go to your browser, search Swiss admin. Okay, search Swiss admin. Then okay, so we can draw our compound here using this function. Just wait for the appearance of the tools. Okay, here this is like draw. Okay, we can calculate any compound easily here, like draw. Okay, and then if we click here this button, this will convert it this compounds smiley file here. So this is also the best way to draw and convert it into its smiley file format. Then if you run this button, click on this button to run, then it will calculate your physicochemical properties. So for example, let's run this one. You also can run individual compound and run at a time more than 10 or many others. So for this compound, let me show the boil egg. So if you click on show boil egg, then the boil egg plot will be here. It is your if you click on the show molecule name, here is your molecule 1. So we insert just a molecule, that's why molecule 1. So molecule 1 is in the range of blood-brain barrier. Okay, so this molecule can pass the blood-brain barrier. Okay, then let's consider its physicochemical properties. Okay, so this is the 2D structure and this is the radar image. Okay, so let me save this image. We can save this radar image. Okay, so lipophilicity, lipo, lipophilicity, flexibility, insaturation, insolubility, polarity, and size. Okay, these all six properties, should be fulfilled. Okay, there are two layers. This is the outer layer, this is the inner layer. Okay, so this layer, this colored layer is the optimal range for this compounds or any compounds. If any compounds just fluctuate from this range, then we can consider this one as uh, just the if the insaturation is violated its standard range, then your compound's insaturation part will be moved uh, in this part. And if the lipophilicity or flexibility also cross the standard range, then also this point and this point will be here. So if your compounds are within this range, then it means that your compounds is standard and in and within the optimal range. So here is your smiley, and let's calculate the physical. Let's consider the physicochemical properties. So this is the formula: molecular weight. So molecular weight is 179, heavy atom 13, and then the number of heavy atoms, aromatic heavy atoms 6. Its fraction carbon sp3, hybridization 0.30, number of rotatable bond 3, number of hydrogen bond acceptor 3, donor 2, molar refractivity 50.31, polar surface area 63.32. So this is the physicochemical properties, and from this, if we calculate the lipophilicity, uh, here is the lipophilicity log P. There, these are the different types of lipophilicity based on different methods. Okay, and here's the consensus lipophilicity 0.36, and all the lipophilicity value are within the range, right? Within the range. So what was the standard range of lipophilicity or log P value? Anyone please? What is the standard range?
Maybe less than 5.
Less than or equal to 5. Thank you.
Less than 1.
What was the standard range for molecular weight?
Less than 500
500.
kilodalton.
Then if we consider the Lipinski's rule or the Weaver rule, is it obey the rules or not? Is this compound follow the Lipinski's rule or not? After considering the molecular weight,
Follows Lipinski's rule.
what about the Weaver rule?
It also follows the Weber rule because the number of rotatable bonds, total number of rotatable bonds are maybe
Less
less
than
than
10,
10.
yeah. And the TPSA or
And also
PSA,
the surface area
yeah,
is less than 140
140.
angstrom square.
Okay, okay. As this compound cross the blood-brain barrier, so in this case, yeah, the TPSA value should be less than 90 angstrom square. Okay. if you are considering this compound for treating any disease related to neurological disorder. Okay, now other physicochemical properties, for example, water solubility. Log S or ESL is one of the method for calculating the log S or water solubility value in terms of log, and it is minus 0.41, which means that very soluble. Okay, it is positive 0.24 according to Ali. So here is the main manuscript for this method. According to this method, this is very highly soluble, and log is in this silicose IT soluble. So according to all the method, our compounds are soluble. Okay, now we can just skip this portion for this time. Drug likeness. Okay, so Lipinski, yes, zero violation, and the Weber, yes, there is also no violation, and other other drug likeness methods. Okay, cause means that molecular weight should be within the range 160 to 480, and log p value less than equal to 5.6, and the molar effectivity is considered here. Okay, okay, now in case of pharmacokinetics properties, for example, absorption, distribution, metabolism, and excretion. So GI absorption, as we mentioned about the GI absorption in our slide. So GI absorption high, blood-brain barrier yes, as we can see from the Boyle method also. So yes, this molecule can pass the blood-brain barrier, and the polyglycoprotein substrate no, and for metabolism CYP, okay, cytochrome these proteins inhibitor are no. So this should be no. Okay, for any molecule this should be no. So the meaning of no in case of metabolism, that's your compounds have no drug-drug interaction chance. Okay, log p, log kp skin permeation is minus 8.16 centimeter per second, and also the medicinal chemistry. If we consider the medicinal chemistry properties like pains and brain, so these two are the methods for based on the structure of any compounds, that if in your structure have any uh, unusual groups or not. Okay, if they have any unusual group, that can be structural problem for the synthesis, or this can produce the toxicity in this compound, then the pains and brain will show alert. So as there are no alerts, so these compounds are properly safe. And according to lead likeness, so these compounds are not like a lead. Okay, because there is one violation in molecular weight. So molecular weight less than 250. So if any compound are considerable as the lead, then we have to follow this uh, criteria. Okay, let me insert all the smiley here. So this time we will insert all these 5 smiley and click on the button run. This will take a few minutes. Okay, you can see the fluctuation of insaturation here. Okay, okay, so for being a lead-like molecule, your compound have to molecular weight within the range 250 to 350. Okay, and a log p value less than equal to 3.5, and number of rotatable bonds less than equal to 7. So to be a lead-like molecule, any compound have to be this criteria. Okay, you can also extract this all results in Excel or CSV file. So retrieve the data as CSV file. You can click on this CSV, then you can okay. Now the question is about the name. Okay, that we just copy the smiley from our this part. You can also copy the smiley from your Excel file. Just 1 minute. So here I have a smiley. Okay, so I have a smiley with my compound ID. Okay, so if I copied the first 10, this means that I copied only the first tens, and I remembered that these are the compounds. Okay, then I can just replace this column with this one. Okay, like this one. So this is just simple. After doing this save this file as XLS. Okay, otherwise XLS file. So here you will find the molecular weight and other properties, and also the water solubility. Okay, this is the log p value. These are the log p value, and these are the solubility. Okay, these are the solubility and GI absorption for our calculated 5 compounds, blood-brain barrier. So only one compound can cross the blood-brain barrier. So yes, you can retrieve the data in your CSV. Okay, for the table preparation. Now we will calculate the toxicity. Okay, so we will use the protox, right? Protox. Okay, so the protox updated to 3.0. So it is the protox. Okay, just click on tox prediction. Just click on this tox prediction. Okay, now you have a option pubchem name. You can search by using the pubchem name or using the chemical smiley. So I'll prefer to search any compound using smiley. Okay, or you can use aspirin and search. Aspirin will appear here soon. Aspirin. So that's why I'm talking about the name difficulty. So let's start with the canonical smiley. Just okay. So here is the compounds, the selected compounds smiley. Now you have to select all. Okay. Otherwise you can select only the organ toxicity. Okay. So let's select all and start tox prediction. In a very short time, it will calculate all the properties related to toxicity. And now let me discuss about the okay. In very first, they calculate the LD50 value. Okay, LD50 value. So it is in milligram per kg, as we mentioned in our slide about the unit of LD50, and the toxicity class 5. So here they mention the class. The class are 1 to 6. So 1 means very toxic and 6 mean no toxic. So the toxicity in case of toxicity class, our compounds are tends to be non-toxic. Okay. Average similarity, prediction accuracy 69.26%. It's okay. Uh, they also calculate the molecular weight, number of hydrogen bond acceptor, donor, and the rotatable bond. Okay, and the polar surface area, log p value. So if you expand this one, then you will show they will show the similar compounds, three similar compounds. Okay, just collapse the toxicity model report. You can also save this as CSV, PDB, Excel. So let me open the Excel file. Okay, so here green color means this is non-toxic. This is the partially toxic. Okay, this is also the partially inactive means non-toxic, but red means toxic. Okay, it is the it is toxic. So you can consider only the hepatotoxicity for your study, uh, like hepatotoxicity, neurotoxicity for organ toxicity only. In case of organ toxicity, okay, you can input this value as the probability score in your manuscript, or otherwise you can use this part, like just you can say this compound are active or inactive. Active means they have the properties or they are showing this type of activities, and inactive means they are not toxic. In case of toxicity endpoint calculation like carcinogenicity, yes, it's not a non-carcinogenic. Immunogenicity, in case of immunotoxicity, it's inactive for mutagenicity. Cytotoxicity, you can calculate the cytotoxicity in case of blood-brain barrier, and then ecotoxicity, clinical toxicity, natural toxicity, the nutritional toxicity. You can just consider this part for your manuscript only. Cytotoxicity, up to cytotoxicity from the hepatotoxicity. Okay, and just add this in a column, the toxicity class and the LD50 value. Okay, you can just search the standard range. Okay, standard range for LD50. Okay, highly toxic substance have LD50 of 5 to 50 milligram per kg. So 50 or less than 50 are considered as the highly toxic, and while partic- practically non-toxic substance have LD50 value of 5,000 to 15,000 milligram per kg. Okay, you can also expand very toxic, moderately toxic compound have the toxicity value 500 to 5,000. Okay, slightly toxic compounds. Okay, and practically non-toxic above 15,000 milligram per kg. Okay, so okay, I think that's all about the pharmacokinetics properties, about the calculation of physicochemical properties, and we also calculate the toxicity. So we can make some table related to our study for our studied compound, and that how they behave like a drug like or not, and what about the toxicity profiling. Okay, here is another tools named as the admit lab. Okay, If you search admit lab, then admit lab 2.0, click on it. You just calculate up to 500 smiley. Okay, you can calculate the admit properties for up to 500 compounds by clicking a single button. Okay, just go to the admit screening, get started. Okay, you can select a file. Okay, you can select a molecule file, you can select text documents, or you can select the SDF, or you can enter a list of smiley. So smiley maximum 500. So I have a compound list, maybe more than 500. Let me reduce this. Okay, 500. Now just paste it here and submit. Just wait a second. Within a very short time it will give you the result. And AdmitLab 2.0 is the updated version of AdmitLab, and also AdmitLab 3.0 may be available. Okay, we consider 499 compound. Okay, they found any, they did not found any invalid molecules. Okay, you can download the CSV file. Just download the CSV file. Okay, here is the result for your 499 compounds admit property. So mm, insert a cell here, and let me copy the ID number. Okay, 500. Just copy and paste. So okay, now you can see all the properties. The log S, log D, log P is the important. Log S is the water solubility. Okay, polyglycoprotein inhibitor or polyglycoprotein substrates. HIA, human intestinal absorption. Okay, CACO2 is also a absorption properties. This is absorption properties. Blood-brain barrier, PPB are the distribution properties. VDS also the distribution properties. These are the metabolism, clearance, and half-life. Okay, and you will find so more properties like LC50. Okay, the lethal concentration is the MS toxicity, right oral acute toxicity, skin sensitivity, carcinogenicity. Okay, right oral acute toxicity and respiratory toxicity. Okay, you can also calculate the toxicity and admit at a time using these tools. And the physicochemical properties. Okay, there is the molecular weight, number of hydrogen bond, acceptor, donor, polar surface area, rotatable bond, number of rings, maximum ring, number of heteroatom. Okay, and then let me show another thing are the LD50. Okay, oral. These are the LD50 oral, and skin sensitization, acute toxicity, right? Toxicophores, genotoxicity, carcinogenicity, mutagenicity, etc. And natural product likeness. Okay, this is the value of natural product likeness. BMS, chelating, PANS, these are the medicinal properties. And here's the Lipinski's rule, Fija, GSK, okay, golden triangle. So These web tools will show the result as accepted or rejected. Okay, you can just short it and you can see the rejected compounds are here. Okay, these compounds are, there are no rejected compounds according to Lipinski. Okay, according to Lipinski. So that's all. And one more thing about these tools, you can also find the you can also view the results in a single for a single compound. Okay, or you can find the explanation here and download explanation page as PDF. You will uh, find the explanation of each properties. For example, what should be the results of toxicology, what should be the result of excretion or metabolism, absorption. Okay, if you go to the tab of absorption, then CACO2, what should be the value for CACO2 permeability and their empirical decision, will it be good or excellent or poor. Okay, so you can just download explanation as a PDF. Here is the PDF file format, or you can read the explanation from this tab. Okay, evaluation for single compound. Okay, evaluation for single compound. Medicinal chemistry, green means okay, red means alert. So in case of toxicity, these are the non-toxic. This shows the non-toxic behavior. This is the moderate toxic, and eye corrosion, eye irritation, respiratory toxicity, they shows the toxicity behavior. Okay, and your LCPT value is here, LCDM. Okay, this is the FM, and the bioconcentration, others. This is the toxin to one pathway. You can the metabolism rate. So plus, triple plus, double plus, and minus. This is the radar image, like suicide me. Okay, this is the radar image. Here is the radar image. They consider lots of properties. Okay, log p, molecular weight, number of ring, etc. So it shows three layer: upper limit, lower layer or lower limit, and compounds properties. So this is the compound properties, and this is the lower limit, and these are the upper limit. Okay, this portion is the upper limit. Okay, I think that's all about the pharmacokinetics properties and drug likeness properties. So thank you for everyone. If you have any question, then you can